Good morning, everyone. Here are my disclosures. So the objectives of our study was to describe the evolution of archived NNRTI-associated resistance mutation, whether or not they were associated with 184V using next-generation sequencing. So we enrolled people living with HIV with more than 18 years, with a prior virological failure to an NNRTI-based regimen. NNRTI mutations were identified in a previous RNA-resistant genotype, and then the patients had an undetectable viral load for at least 1 year with no blips before the next samples and the next genotypic test on DNA. And it's important to note that at this time, at DNA1, patients were undetectable for a median time of 7 years. Then 2 blood samples were available for NGS analysis, DNA1 in 2019, and the second one 5 years later in 2024. So we used Illumina technology with a 5% threshold for the analysis, and any NGS was successful in 96% of the samples. So overall, we had genotypes in RNA with different kind of NNRTI mutations, such as 103N, 106, 181, 188, 230, and among the 59 patients, 40 had the 184V in addition to the NNRTI mutations on the RNA genotype. So as you can see, we studied 49 people living with HIV harboring NNRTI mutations in the past. And we, when we observed the results at the first point on DNA, 55% of these patients had no NNRTI resistance mutations on the genotype, and 55% have still at least one NNRTI resistance mutations in the DNA. So you can see on the graph that overall the number of NNRTI resistance mutations in and 184V observed were lower in the samples at DNA1 than in RNA, and this is true for all the studied mutations. So we looked at the factors that were associated with the disappearance of the mutations in DNA1, and among the factors we studied, the only factor that was associated with NNRTI resistance clearance was a shorter duration of NNRTI replication at virological failure. So patients who clear the mutations at 37 months of median of replication on NNRTI versus 5 for the other. And we observed also that there was a trend towards an association between NNRTI resistance associated mutations clearance and a longer duration between RNA and DNA, that 14 years versus 9 years. If we look at the coevolution of NNRTI resistance associated mutations and the 184V at DNA1, you can see that among the 40 patients who had the 184V in addition to the NNRTI resistance mutations, the 57% of patients had no NNRTI RAMs, and it was the same with the patients who did not harbor the 184V in addition to the NNRTI RAMs. So the presence of historical 184V does not seem to impact the clearance of the NNRTI-associated mutations at DNA1. And we observed the same results when we compare the results in DNA2, the second sample. Finally, we looked at the mutational viral load in HIV DNA, and as you can see, there was no the HIV viral load was similar between DNA1 and DNA2, and there was no clear evolution of the viral load for all the mutation studied, NNRTI or 184V. So in conclusion, overall, the number of NNRTI and 184V mutations seem to decrease over time in the reservoir. The presence of historical 184V does not seem to have an impact of the reduction of NNRTI RAM in the reservoir, and the only factors associated with the persistence of NNRTI resistance in the first DNA sample was the duration of replication under an NNRTI regimen. So probably we did not observe, we observed a first decrease of the mutations and then a kind of plateau, probably because the patients we studied were already suppressed for a long time, 7 years in median. Thank you for your attention.